Hello, everyone. Welcome to the 5th day of the Vaccinology course, which is also the last day. My name is Birkinet Adesa. I'm a research scientist at the International Vaccine Institute's Epidemiology Public Health Impact Unit. I will be moderating day 5 sessions on behalf of Dr. Florian Max. Today's sessions will be on antimicrobial resistance, epidemiology, and health economics. And we'll have 5 presentations lined up for today. Before we go to the presentations, I'd like to share a few housekeeping rules. The first one is recordings of today's sessions will be available on IVI's YouTube channel at about 3 p.m. Eastern Time. The recordings will only be accessible during this week, so please go ahead and watch them. Today's lectures will be followed by a live Q&A session. Please submit your questions in the Zoom Q&A platform. Please also kindly note that we may not have enough time to address all the questions live. Generally, course observers will review and refer your questions to lecturers, and they will be responded to as time permits. The presentation slides will also be shared at the end of the course. As in previous days, a short poll will be sent out at the end of every lecture. The polls are anonymous, and your feedback on the lectures and the speakers will be very essential to develop the course for future vaccinology courses. For those who are joining us live on Zoom, there will be a digital certificate that will be awarded to those who have attended more than 90% of the sessions during the whole week. We will be reviewing your login details to assess if you are eligible or not based on the participation. Having said that, we'll go to our first presenter for today, Dr. Elisabeth Klemm, who is a senior research advisor at the Wellcome Trust. Her presentation will be on the role of vaccines in reducing antimicrobial resistance. Dr. Klemm, please.
Hello, my name is Elisabeth Klemm, and I work at the Wellcome Trust. Today, I will be talking about the role of vaccines in reducing antimicrobial resistance. AMR is a serious and growing problem, and it is already responsible for 700,000 deaths per year. This number is expected to rise to 10 million by 2050. AMR is the next big public health crisis looming on the horizon, and we are slowly and steadily walking towards it. But there are tools available that we can use to lessen and delay the problem of AMR, and today I will be talking about one of those tools, which are vaccines. I will first go over the different ways that vaccines have an impact on AMR, including the concept in general, and then presenting some specific examples and data. After that, I will discuss the opportunities for developing new vaccines that could fight against AMR. And I will finish by going over the next steps to put these things into action, using the WHO's action framework as a guide. AMR is a very complex problem with multiple contributing factors. Therefore, it is important to seek out and use multiple solutions as part of a multifaceted response. Vaccines are an important tool to be used alongside other approaches, including improved water, sanitation, and hygiene, better diagnostics and stewardship, and the development of alternative therapeutics and new antibiotics to replace those that are no longer as effective. Vaccines, by their nature, are able to offer a long-term approach, especially when compared to antibiotics. This graphic shows the years that new antibiotics and vaccines were introduced and when resistance was first observed. You can see on the top half that resistance to antibiotics emerges quickly, often within a decade, while for vaccines, the time to resistance is much longer, if at all. And we are still using vaccines that were developed several decades ago. There are notable exceptions to this rule, including the influenza vaccine, which needs to be redesigned each year to address new mutations, and the pneumococcal vaccines where we have seen serotype replacement. When we are considering why we readily see resistance emerge for antibiotics and not for vaccines, we need to think through the differences in when and how they work. Vaccines prime the immune system so it is able to rapidly and efficiently clear the pathogen soon after exposure. But for antibiotics, the bacteria is able to multiply in the host before treatment. During this population expansion, the bacteria may acquire mutations and mobile genetic elements, some of which may confer resistance. When the antibiotic treatment is administered, the bacteria that has developed resistance to that antibiotic will survive and can be passed on to other hosts. The key differences here that are thought to make vaccines less prone to resistance are the timing and the target. For the timing, the vaccine-induced immune response acts early before the bacteria has had a chance to multiply and mutate. For the target, many vaccines include multiple antigens, so it would be very unlikely for a pathogen to acquire mutations in all these targets at the same time. So in which ways do vaccines contribute to tackling AMR? There are three main ways. The first is by directly preventing the infection, carriage, and transmission of drug-resistant organisms. The second is by reducing the occurrence of symptoms that lead to antibiotic use, and thereby providing selective pressure for the development of antibiotic resistance. And the third way is by preventing secondary infections with drug-resistant organisms. It is very clear to see how vaccines that target pathogens which harbor AMR themselves fulfill these three roles. But it is also important to acknowledge the role that vaccines against non-AMR pathogens play in fighting AMR. This includes vaccines for viruses, such as influenza. While they don't directly prevent drug-resistant infections, they reduce significant amounts of antibiotic use and secondary infections. This schematic goes into more detail to illustrate the effects that viral pathogens can have on AMR and how they can be prevented with vaccines. As shown, an individual gets infected by a virus, and although this is not a drug-resistant infection, it still results in symptoms like fever or diarrhea that could lead the patient to seek out antibiotics. In this case, the antibiotic use would be considered inappropriate, since the antibiotic won't help with the viral infection. Additionally, the patient may develop a secondary infection with a bacterial pathogen. This bacterial pathogen may require antibiotic treatment and may be drug-resistant and could be transmitted to others. But if the individual is vaccinated against the causative agent of that secondary infection, then this will be prevented. And if the individual is vaccinated against the viral pathogen, then all of this will be prevented. Secondary infections can be frequent for respiratory pathogens. During the COVID pandemic, doctors were giving antibiotics frequently to prevent secondary infections. In this meta-analysis from the first 6 months of the pandemic, nearly 3/4 of patients were given antibiotics. As it turned out, bacterial co-infections were relatively infrequent for COVID patients, but doctors were still prescribing antibiotics, especially in severe cases. Now that we have a COVID vaccine available, we would expect to see the amount of antibiotics prescribed to decrease. I would now like to share a few examples of studies that have looked at the impact of vaccines on AMR, and specifically at how vaccines have been shown to decrease the occurrence of drug-resistant infections. It has been shown repeatedly in several different countries that the introduction of the pneumococcal conjugate vaccine, or PCV, results in the decrease of invasive pneumococcal disease caused by drug-resistant strains. PCV was in fact designed to include serotypes that were known to be drug-resistant. Here we have data from the USA on the left and from South Africa on the right. In both countries, there were significant reductions in the cases of invasive disease with resistance to the different classes of antibiotics as shown. The next sets of examples that I would like to share look at the reduction in antibiotic use by vaccinated individuals. This first example comes from a study that analyzed data from low and middle income countries. The researchers looked at pneumococcal disease and found that children under 5 years of age who were vaccinated with PCV had a decreased odds of receiving antibiotic treatment for acute respiratory infections. The reduction was especially evident in 2 to 5-year-olds with an almost 20% decrease. As a control, the researchers looked for changes in antibiotic treatment for diarrheal endpoints linked to PCV, and as expected, did not find a significant change. Overall, it was estimated that PCV in these countries accounts for a reduction of 23.8 million antibiotic-treated episodes per year. The same group also analyzed the data for the rotavirus vaccine and saw an 11.4% decrease in odds of receiving antibiotic treatment for diarrhea in children under 2 who were vaccinated with the rotavirus vaccine. A third example comes from an observational study in Canada that looked at the influenza vaccine and changes in antibiotic prescriptions across different provinces. The province of Ontario made the influenza vaccine universally available. It was shown that the number of antibiotic prescriptions in that province decreased by 64% compared to the previous year, whereas there was not significant changes in other provinces that had not made the influenza vaccine universally available. The final example is from aquaculture in Norway, where they started using a vaccine against furunculosis for salmon farming. The amount of antibacterials in tons is plotted with the blue bars in the graph at the right, and you can see a sharp drop-off in the early 1990s when the vaccine was introduced, while the production yield, which is shown with the green line, went up. Vaccines in agriculture and aquaculture are an important way to remove significant amounts of antibacterials from the system. In this case, we're measuring in tons, and so it's important not to forget One Health. I'd like to add another thought on the impact that vaccines could have on antibiotic prescribing behavior beyond the target pathogen. This example is for typhoid fever. Fever is often treated empirically with antibiotics in areas where typhoid is endemic. But in reality, the majority of febrile cases are more likely to be due to viral infections and thus should not be receiving antibiotics. The data at right shows that between 3 and 25 patients receive antibiotics for suspected typhoid for each true case of typhoid. The doctors are prescribing for the worst case scenario of typhoid because they don't want to miss those true cases. But what if the typhoid vaccine is able to reduce or eliminate that worst case scenario? Doctors would be less likely to automatically give antibiotics for fevers. The same could also be true for strep A, which is the leading bacterial cause of pharyngitis, and we currently don't have a vaccine available. These examples I have shown come from individual studies, and they go a long way to establishing the role that vaccines play in reducing AMR and antibiotic use. 2 recent systematic reviews have been published, which look specifically at vaccines and antibiotic use, and these allow us to take stock of the entire evidence base and to make quantifiable estimates across multiple studies. 96 studies were identified, of which 24 were RCTs. Both of the systematic reviews concluded that there is support for the hypothesis that the pneumococcal and influenza vaccines reduce antibiotic use. But differences in the study design, the data reported, the types of vaccines that were used, and the populations they were tested in made it difficult to draw broad, definitive conclusions. Furthermore, it was noted that there was little data from LMICs and for vaccines other than PCV and flu. These are important evidence gaps that, when addressed, will allow policymakers to make better informed and nuanced decisions around the use of vaccines to fight AMR. I will now move on to the second part of my talk, which is discussing opportunities for developing new vaccines that would help fight AMR. The WHO has published a list of priority pathogens for the development of new antibiotics. These are all bacterial pathogens that are currently exhibiting increasing resistance. There is no similar list of priority pathogens specifically for the development of new vaccines to fight AMR. When we are thinking about which pathogens we should consider for vaccine development in the context of fighting AMR, we should also include pathogens that contribute to antibiotic use. For those pathogens where there is already a licensed vaccine available, the emphasis should be on collecting data on its impact on AMR and increasing coverage of the vaccine. But for those pathogens without an effective licensed vaccine, we need to first think through the questions of what impact a vaccine would have on AMR, whether it's feasible to develop a vaccine, and whether the vaccine would be implementable. Answering these questions will help to provide us priorities of which vaccine should be developed. We at the Wellcome Trust commissioned the Boston Consulting Group to answer these key questions for those pathogens that are on the WHO AMR priority pathogen list, and to determine which would be most suitable for vaccine development. This report was launched in October 2018, and it's available at vaccinesforamr.org. The evaluation assessed each of the priority pathogens across 3 dimensions. The potential health impact was analyzed by looking at the disease burden, mortality and morbidity, the urgency of the AMR threat, and this included how treatable it currently is and the trends of resistance, and the attributable antibiotic use. The report also evaluated the probability of R&D success by looking at how many candidates were at different stages in the pipeline, as well as the pathogen biology and ease of preclinical and clinical R&D. The probability of uptake was also evaluated. This is to answer the question of, if a vaccine were developed, would it be used? The analysis included the expected policy stance, the likelihood of payer, government, and Gavi support, and any barriers to uptake and commercial attractiveness. The results of the evaluation are summarized in this matrix, with the health impact on the y-axis and the probability of R&D success on the x-axis, and the incidence reflected in the size of the bubble. The pathogens were clustered into 4 groups, with recommended prioritized actions specific to each cluster. The first group consists of pathogens for which there is already a vaccine on the market. For these pathogens, action is needed to increase access and uptake. The second group is pathogens where there is a good understanding of the pathogen, and it was recommended that new vaccines be brought to market by accelerating clinical development. The third group of pathogens had a less clear path for R&D feasibility, and so it was recommended to invest in early stage research to advance early R&D. The last group of pathogens were deemed less well suited to vaccine development for various reasons, and it was recommended to explore alternatives and/or collect more data on burden and transmission. This brings me to the final part of my talk, considering next steps. The WHO released an action framework on leveraging vaccines to reduce antibiotic use and prevent AMR earlier this year, and it served as an annex to the Immunization Agenda 2030. In it, there were 3 goals: to expand the use of licensed vaccines to maximize their impact on AMR, to develop new vaccines, and to collect and share more data on the impact of vaccines on AMR. For this first goal of expanding use of licensed vaccines, it is worth looking at the current global coverage levels. These numbers were taken from the WHO calculations in 2019. As you can see, We're well below where we could be, and an increase in coverage would help to combat AMR. For PCV, we're at around 48% global coverage, but if we achieved universal coverage, it is estimated that that would reduce antibiotic treatment by 11.4 million days per year. So this brings me to the end of my talk, and in conclusion, the key takeaways are that vaccines play an important role in AMR by reducing drug-resistant infections, reducing antibiotic use, and reducing secondary infections. And vaccines are already having a huge impact on AMR, but we can do more to maximize that impact if we expand the use of licensed vaccines, develop new vaccines, and collect more data on the impact of vaccines on AMR and antibiotic use. Thank you so much.
Thank you, Dr. Klemm, for a wonderful presentation. We have a couple of questions from the audience. And the first one is, it's very important or essential that vaccines can prevent AMR. Could it be possible for some bacteria to evolve to evade the immune system like viruses do? That's the first question we had from the audience.
Thank you for the question. Yes, it's always true that there can be new mutations that could overcome the vaccine. This is more likely when there are vaccines that are targeted at just one antigen. So, for example, the diphtheria vaccine is only against the toxin, and we have seen some new variants, and we don't know if those are linked to lower efficacy of the vaccine, but yes, we would see changes over time and mutation. That's what bacteria is very good at doing. But it tends to be that when there are multiple targets and antigens that are included in the vaccine, it's harder for the bacteria to change all of those at once. So if you have a vaccine that is looking at multiple antigens, then if one changes, the other will still be there and have those antibodies for protection.
Thank you. And the second one is specifically on tuberculosis and vaccination. And the question really says, despite BCG vaccination, now there are emerging drug-resistant tuberculosis cases in the developing world. And what could be the reason behind that is the first question. And then the second question follows up on like any potent new vaccine that for tuberculosis specifically.
Yes, those two questions are linked, and definitely there is a lot of XDR TB that we're seeing, and this has long, difficult treatment times with antibiotics. The current vaccine, BCG, that we have is not effective on pulmonary disease in adults in preventing that. So you're still getting a lot of transmission and spread, which will mean that we have exposure to antibiotics and development of resistance. So that's always going to be happening with the current BCG vaccine. There are other vaccines that are in development that are thought to be more effective, and so we're hoping that those will continue to be developed and maybe be better than the BCG vaccine and answer some of these problems.
And we also have a third question, and it's about the relationship between the use of vaccines and resistance against other pathogens, such as viruses, parasites. Would there be any relationship between the use of vaccines and resistance against other pathogens, if they impact each other in any way? Yeah.
I don't quite understand the question there. Sorry.
It's basically using vaccines for one pathogen and I think impacting resistance in other pathogens, for example, in parasites, let's say, or viruses, yeah.
No, I don't think that's been observed, and I wouldn't expect that to happen since vaccines would be quite specific to their target.
Yeah. And another question is more about the governance. How best can governments be involved in tackling AMR, especially in the context of vaccinations?
Yeah, I think that the government has a big role to play in this. A lot of governments have been urged to put together AMR national action plans, and these are great steps to bring forward on the fight against AMR. It's important that these action plans recognize the role that vaccines play and include them in their plans to increase coverage of vaccines if they're not already offering all of the vaccines that we have that are known to prevent AMR, to increase that coverage and make sure that it reaches all of the populations. Also in those countries that could invest in the development of new vaccines.
Thank you. I think maybe one last question is about how we measure actually the impact of vaccines in reducing antimicrobial resistance. What would be the most important or the most convenient approach in terms of evaluating how vaccination actually could impact antimicrobial resistance?
Thank you for that question. This is a very important thing to make sure that we have the right data. The two key things to measure are the level of drug-resistant infections before and after a vaccine or in an RCT, and then also the antibiotic use. So there's lots of different ways to measure this, and it's important to be able to compare them across studies. So if there's looking at the data that's been published, we see it in difference of prescriptions and days on antibiotics.
Thank you so much, Dr. Klemm, for answering to each of the questions. And please feel free to provide the responses live in the Zoom Q&A platform if there are any unanswered questions. Thank you. And with this, we move to the second presentation. Our second presentation, which is by Dr. Iruka Okeke, who is a professor of pharmaceutical microbiology at the University of Ibadan, Nigeria, will be on whole genome sequencing on in antimicrobial resistance. And over to you, Dr. Okeke.
Hello, everybody. My name is Iruka Okeke. I'm a professor at the University of Ibadan. I study enteric pathogens, and I'm particularly interested in diarrheal pathogens, typhoid and enteric fevers, and antimicrobial resistance. Today, I'll be talking about sequencing in antimicrobial resistance, which is the topic of my presentation. Less than a decade ago, there was very, very little data from low and middle income countries, particularly those in Africa, which is where I live, that provided any information at all about antimicrobial resistance. And this is a problem because if you want to address antimicrobial resistance, you need to know the scale of the problem, and you also need surveillance data to be able to monitor progress when you implement interventions. Now, to rectify this, the WHO wrote a global plan on antimicrobial resistance and also mandated in 2015 all countries to come back in 2017 with national action plans on antimicrobial resistance. Now, just for those of you who may not be aware, a national action plan typically has 5 pillars, and one of these pillars is surveillance in humans, but also in animals and in the environment. So you had a situation where countries like mine, I'm from Nigeria, actually had to very, very quickly, if they didn't have plans in place already, had to very quickly put together a plan. And that plan had to include a surveillance component. And it's often best to have this plan, or at least a surveillance component, led by a public health institute. We were fortunate then to have a very dynamic leader of our Nigeria Center for Disease Control. And he put together a group that wrote the National Action Plan, as well as was mandated to develop a surveillance system. So now developing a surveillance system can take a while. I've been in lots of communications with the Philippines, for example. They have a very nice surveillance system for a middle-income country, and it took them over 30 years to build it. And we didn't have 30 years. I mean, in 2014, there were no data, and we wanted to have data as soon as possible. So that means that what we really have to do is to look at new technologies now that could help us leapfrog over the more difficult things that are needed to build a surveillance system. And one of those technologies happens to be whole genome sequencing. Now, some of the roadblocks that we have to deal with are not just being able to gather isolates from isolates from clinical specimens and test them for resistance, but this needs to be done with a number of quality parameters that many of our labs don't have. So actually building a surveillance system gave us the opportunity to improve quality management across diagnostic labs for bacteriology in ways that might have been a bit more challenging. And again, this is an area where whole genome sequencing has helped. So when we wrote our national action plan, we actually presented it in 2017. One of the things we wrote into the plan, and we actually criticized for it, was that it would have a genomic sequencing component. We were criticized not because anybody believes that genomics is bad, but simply because genomics is expensive. The costs were higher then than they are now, and it was believed that there were ways to do this more cheaply. However, when we looked at all the options, we realized that it would actually take longer and probably cost more to use the traditional mechanisms to provide that level of quality support to the antimicrobial surveillance system. So we felt, and I'll show you some examples, that having genomic surveillance from the start would help leapfrog us over some of those hurdles. And then a big hurdle that we had was that we didn't have a reference lab that was sufficiently resourced to be able to provide the types of services that antimicrobial resistance surveillance would need. Now, for surveillance of antimicrobial resistance, unlike for many other types of surveillance, you're working with many different types of bacteria. So a reference lab has to be able to subtype all of these many different types. And so being able to build that capacity quickly would be challenging. However, whole genome sequencing offers us the opportunity to be able to do that across the board for different organisms. And then in addition, it would support things like outbreak investigations. Whole genome sequencing would help us to be able to see when we had resistant clones spreading in one part of the country, whether they were the same as clones that were in other parts of the country, or even in other countries in the world. And then because those of us who are helping build the surveillance system were largely based in academia, in order to keep us engaged, it was nice to be able to nest research questions within the surveillance system. And whole genome sequencing made it very easy to be able to do this. So what do you need to actually have a whole genome sequencing component of an antimicrobial resistance surveillance system? First of all, you need really good diagnostic labs. You need really good Sentinel labs that are going to collect samples, process them, get the isolates, and then you need obviously a coordinating center that is going to coordinate the whole process. This is what the Nigeria Center for Disease Control is doing for us. And then you need to make sure that your Sentinel labs are not just Sentinel labs, that they're Sentinel sites that are collecting requisite metadata that can be paired with the isolates. Isolates can be sent to reference sites for sequencing. When the reference site receives them, typically they will validate the isolate, which provides some quality information for the Sentinel labs, extract the DNA, and then assess the DNA, make a library that can be used then by the sequencing machine to sequence the DNA. Once the DNA has been sequenced, this is not the end of the process. We find that very often people think that, well, once the data comes out of the machine, you're done. You then need to be able to analyze the data. This is something that's typically done by bioinformaticians in order to figure out what it means and how it can be used and what it's telling you about the resistant organisms. Now, just to say that at this point in time, the data that you get from whole genome sequencing is not sufficient to guarantee resistance or susceptibility. So what that means is that surveillance systems such as GLASS, the Global Antimicrobial Resistance Surveillance System, are requiring phenotypic data that has measured whether or not the organisms are resistant. Genomic data will tell you what the resistance mechanisms are, but sometimes you can have a resistance gene that's not expressed, in which case you'd have information in the genomic data that says resistance is potentially there, but it's not actually there. And other times, although much less commonly, you could have a new resistance mechanism that you would not be able to recognize using traditional bioinformatic pathways. And for that reason, it may be missed. So right now, even if you have genomic surveillance, typically you're going to have to pair this with phenotypic testing in your surveillance system. So some of the challenges with setting up a genomic surveillance component to an AMR surveillance system, obviously, is that you need to set up the laboratory resources that are going to be able to do this. This does not only involve buying the machines, but also making sure that the machines are housed in situations where they will work. They need continuous electricity power. They need a certain temperature and humidity requirements, which may not necessarily be the ambient ones in the country that you're working with, certainly not if it's a tropical country. Obviously, you need to have the equipment set up. You need to have it serviced. Unfortunately, many of the sequencing systems that are used today are run out of monopolies, and they may or may not have good representation in different countries. So some of this can be a challenge depending on where you're working. And then obviously you need to be able to build human resources, not just to do the sequencing, but also to do the bioinformatics that allows you to interpret your finding. And this comes with the need for very robust procurement and financial management systems, because sequencing reagents and equipment tend to be quite expensive. The supply chains, initially, you may have to set them up if they don't exist in your country. So having that level of administrative support can be very helpful. So let me give you some examples of particular areas where genomic surveillance is very valuable, particularly in low and middle income settings like mine. One of them is that you can actually replace the requirement for some reference lab services by using whole genome sequencing. When we started out, we found that many of our labs were actually quite good in doing, many of our candidate labs were quite good in doing culture and susceptibility testing. But we're not very good in being able to reliably subtype. And we didn't actually have a reference lab that could do this reliably for the system. However, genomic sequencing allows you to subtype isolates very finely and to do so with considerable accuracy. Okay, let me give you an example of why this is important. And this is data that is actually out of Nigeria's surveillance system. So we noticed that quite a few of the Sentinel labs were sending us Enterobacter species. Most of the time when they sent these Enterobacter species, they were not identified as Enterobacter. A few of them were, those that are in blue, but many of the times the labs did not have sufficient reagents and identification systems to identify them as Enterobacter. So they were identifying them as the family. These were just Enterobacteriaceae. And a few of the labs were misidentifying them as Pseudomonads or as Acinetobacters or something else. Now, the reference lab was using VITEC to identify the organisms, and so had a better identification system than the Sentinel labs that are using tube biochemistry. Still, they were finding that many of the strains could not be speciated. They were part of the Enterobacter Hermetier complex, and the page I'm showing you right now are strains that eventually turned out to be a species known as Enterobacter Hermetier. If you haven't heard of it, you're not alone. It's not that common. But we find that it's actually quite common in our country, and it's something that we actually did not know. And it's something that if we had not had whole genome sequencing as part of our reference lab process, that we still would not be in the know about, because we would have just classified these as Enterobacter Hermetier complex. Also be aware that the VITEC is not correctly identifying all of them. Now, not only can we identify them as Enterobacter Hermetier, but we can actually subtype them into sequence types, again, using whole genome sequencing. Even within the sequence types, we can tell which ones are clonal from whole genome sequencing. So you have very fine level of subtyping. And you also are have very good and fine level of identification, which is very important for a reference lab to have in a very good surveillance system. Now, something else that whole genome sequencing can help do is to help investigate outbreaks of resistant bacteria. There are lots of different ways to do this. Okay, you can use lots of different subtyping schemes to be able to realize that organisms are clonal, and then if they are from the same time and place, that they might be part of an outbreak. But sequencing makes it very easy to do this. Okay, and I'll give you an example. One of our Sentinel labs had reported that they had a likely outbreak of Acinetobacter in their ICU. And they sent us the strains. We also had strains that were sent by them and by other sites that were also Acinetobacter. We sequenced them all and then made a phylogenetic tree based on the sequencing. And the strains that were sent as part of the outbreak here are marked A. Okay, and as you can see, not just from the tree, if you're good at reading trees, you can see that the organisms appear clonal, but also from the data that we're able to get from the sequence that tells us which resistance genes, these are all resistance genes, sequence types, and so on. You can see that these strains are essentially identical. And so the resident that drew this to our attention, Emmanuel, was actually right. There was an outbreak. However, what he did not know was that he had two outbreaks going simultaneously in the same institution. Okay, one is not from the ICU. And this is, again, something that we could find by doing whole genome sequencing of the isolates that are being referred to the reference center. Now, this was a case of somebody noticing something in his ICU and drawing our attention to it and requesting support. But you can also have a situation where this could be identified by the reference lab, even if it was not recognized by the Sentinel. I just want to show you some examples from Klebsiella. Klebsiella, as you know, is like Acinetobacter, is a WHO priority organism for AMR, and it's very concerning that the number of resistant Klebsiella in Africa being reported in the literature is rising dramatically. One of the reasons why it's rising is that more people are doing surveillance, so there's more activity in this space. But we also feel that resistance is becoming more common. This is a case study that showed that in a particular case, not being able to identify the resistant organism, which turned out to be an ESBL producing Klebsiella, led to a huge financial cost for keeping the patient alive until finally there was susceptibility testing. The susceptibility pattern was revealed, and then the patient was treated with the right antibiotics. So having good Sentinel labs, good diagnostic labs is important. But look at what happens when they contribute to a surveillance system that has genomic support. So this is a phylogenetic tree of Klebsiella pneumoniae from Sentinel sites within the Nigeria surveillance system. Okay, and this tree is like the trees you've seen before, except this one is presented to you as a circle because we've got many, many leaves on the outside. And anything that has a horizontal line that is almost straight is suggestive of a clone. That's something, an identical organism that's spreading around. And you can see that we have quite a few clones. And if you look at the leaves of the tree, the tips, you'll see that we've colored them by the institution that is sending them. And what you can see is that there's an institution in red that's sending a lot of Klebsiella, but also has a lot of clones. And so we looked into each of these clones separately to ask what happened. Okay? Now, in addition to looking at the tree, you can also see that there's a general lack of diversity in this particular institution, OAUTHC here, where you can see that the number of O types of Klebsiella, this is the O types are the LPS type, is relatively small for the number of isolates that they're sending, which suggests that there's less diversity. The same is true for K types, capsular types. And so we asked them, are any of these infections likely to be hospital acquired? And they said yes. They sent that information in, and you find out that the sequence types where we're seeing a lot of samples are actually the ones where they had a lot of hospital acquired infections. I'm blowing up a few of them for you. Not all of them turned out to be outbreaks, but a lot of them did. This is sequence type 307, which is the most common in Nigeria. So it's not odd that we had very many of them, and you can see from the leaves that they're found in all the different sites. But you can see this cluster A here, which is essentially an outbreak that occurred. It was within the span of about about 3 weeks. These isolates were recovered, and the resistance patterns were very similar, and the isolates were clonal. This is an outbreak that happened, and the hospital was not actually aware that it was an outbreak. This is a second outbreak of Klebsiella in the same, a second and a third outbreak of Klebsiella in the same institution. And again, you see you've got clusters of resistant organisms that were isolated, in this case, in a wider period, about 6 weeks, so that they were not able to notice that within those 6 weeks, there were these organisms that were clonal, because there were other organisms coming in at the same time. Whereas sequencing clearly showed that the organisms are clonal, they have similar resistance patterns, and so they must have been transmitted within the hospital. And so what this required is for infection control in their institution to take a closer look at their practices and do something so that we can reduce the frequency of outbreaks within that institution. Again, this is something that could have been done without genomic surveillance, but genomic surveillance makes it much easier and actually less resource intensive to investigate all of these outbreaks. I did want to point out that if you're doing genomic surveillance within your antimicrobial resistance surveillance system, you have the opportunity to gather additional information that could have other uses. These include surveillance for important pathogens like those that cause cholera, bacillary dysentery, typhoid, and so on, but also the ability to get information that might inform vaccine development. So this is just a pipeline of how a bioinformatician processes Klebsiella data when it comes off a sequencing machine. And most of it is for figuring out whether the organisms are clonal and figuring out what antimicrobial resistance genes they have. But he can do this simple step with Captive just by clicking his mouse, and he can get O and K serotypes of the Klebsiella isolates. Now, we don't necessarily need this because we have other information that is finer about clonality. But one of the reasons why it's really useful is that it informs vaccine development. K antigens, capsular antigens, and O antigens, LPS antigens, are actually highly immunogenic. And so they're potential targets that those who are developing vaccines are looking at. The problem is that there are very many of them, okay? There are hundreds, certainly 100 and something of each. So those developing vaccines have to decide which ones will go into the vaccines. And as you can see, here's a global map of the distribution of O types. O types actually hold the most promise because they do less switching. And what you can see is that there's not that much data from Africa. And the data are from a few countries, and the data are different from those countries. So you can see that although O type 1 is very common, O types 2 or 5 may be the next common depending on the country. And also, if you know where Nigeria is, which is right here, you can see that there's no data from Nigeria. So now we have data just because we're using sequencing for AMR surveillance, and we can predict O types and K types from the sequence. And so we now know, for example, that O1 is the most common in our country too, and O2 is the next most common, but O5 is very common. So this is something that we can get without making additional investments from our country because we have whole genome sequencing. I would have shared more examples, for example, cholera examples, typhoid examples that are in the literature. Take a look at them. I just felt that in the interest of time, that I should share some of the things that may be less obvious about the benefits of having a sequence-based surveillance system. Now, I'd like to just end by letting us know that sequencing in a surveillance system, while we're using it in a reference lab context, that this is not the only space where it would be useful. Okay, it was certainly a space for us to start, but even Sentinel labs could get a lot of information and improve their services if they had local sequencing. And down the line, it's certainly something to think about. For example, outbreaks could be identified in almost real time if the sequencing was being done at the site where the infection was actually happening. Additionally, new technologies that are coming on board may allow us to sequence directly from specimen and therefore reduce the turnaround time of processing bacteriology isolates. So it's certainly a space to watch. There are a lot of different sequencers. Many of them are becoming more affordable. There's a handheld MinION, which can be used at with very little upfront costs at small sites that may not want to do a lot of sequencing. Okay, I hope I've been able to show you that sequencing has a very important role to play in AMR surveillance. If you are from a low-income country in Africa and you don't have sequencing, and you do have a surveillance question to ask, there is a network that is supported by the Fleming Fund known as Seek Africa, of which we are part, to which you can submit proposals and isolates to and have them sequenced. You can do your own analysis if you like, or Seek Africa can do the analysis for you if you don't have bioinformatics capacity. And Seek Africa also offers training in order to build this capacity if you're thinking of introducing sequencing in your country in the future. Okay, I'm going to stop here. I'd like to thank those in my lab that are involved in sequencing, including those that I've grayed out because they've moved on. I'd also like to thank the funders and the organizations that have helped us set up genomic surveillance in Nigeria and help us to use it in genomic surveillance. I'd like to thank you for your attention, and I'll be happy to take any questions that you might have. Thank you very much.
Thank you very much, Dr. Okeke, for the very informative presentation. We have some questions from the audience, and I will read them for you. Thank you for joining us online. The first question is, it's clear that whole genome sequencing has value in surveillance. What would be the role, or does it have a role in clinical management of patients? Is the first question, Dr. Okeke.
It's a really good question. The answer is that it certainly can. There's a group at Adam Brooks Hospital in Cambridge that's actually doing this. The important thing is that if you're going to use whole genome sequencing for managing patients, you have to really shorten the turnaround time. So unlike what we are doing, which is providing reference lab services, the turnaround time for us is quite long. But if you have sequencing at your own facility, you can shorten that turnaround time considerably. And if you can do direct from specimen sequencing, for which methods are just being developed, you can even shorten it so that it's shorter than traditional bacteriology. So there's a lot of potential for doing this routinely in the future. But right now, it's only being done at specific sites that have these technologies or are developing them.
Thank you. The second question we have is, although whole genome sequencing may help to fight increasing antimicrobial resistance, access to sequencing itself has been a barrier in addition to infrastructure, human resources to analyze such big data. In understanding the benefit of whole genome sequencing over the traditional methods, has there been a study that compared cost-benefit of whole genome sequencing compared to other methods or like the traditional method? If not, do you think whole genome sequencing should be prioritized in certain contexts over the traditional surveillance system? Yeah.
Okay, thanks for that question. So we definitely need more research on cost effectiveness. There's been some studies, notably in the UK, that have found that whole genome sequencing methods are more cost effective for reference lab services, to the extent that Public Health England has actually moved their Salmonella reference services to whole genome sequencing. So that has happened because it's more cost effective. There's also data from the UK that shows that the ability to identify outbreaks makes whole genome sequencing a really effective tool, cost-effective tool for infection control. Now, what we really need are data from low and middle-income countries. I am one of those interested in collecting those data. Unfortunately, for many of us, two things. First of all, for many of us, whole genome sequencing is very new. So it's going to be a few years before we can amass enough data. We know that the upfront costs are very high. What would really make it cost effective is that the running costs become lower over time. And then the second reason why getting cost effectiveness data is a bit of a challenge is that costs for whole genome sequencing are unfortunately much, much higher in lower middle income countries because of our supply chains. And so we have to figure out if that changes, how will cost effectiveness change? And then thirdly, I should just mention that if you put in the cost to reducing mortality, reducing morbidity, the cost effectiveness jumps. But the ways to measure this in lower middle income countries, one has to ask questions about whether or not those are the correct ways to use for this. So essentially, there's interest in collecting those data. I suspect that it's cost effectiveness, but the hard data that's going to show it isn't out yet.
Thank you so much. Yeah, the next question is, it really says, AMR is a current major problem currently. In the case of tuberculosis, drug resistance is one of the most challenging problems, especially in sub-Saharan Africa and Southeast Asia. Is there any applied whole genome sequencing surveillance system for such specific case to tackle these challenges?
Yeah, whole genome sequencing for TB surveillance actually preceded AMR surveillance. As you rightly suggested, it's really important in that context. And actually, that's one example I could have given where it's very important in care because the care is so much more drawn out. I know that there are surveillance systems. I'm not that familiar with them. It's just not my area, but certainly search for them online. They exist, and they've actually developed a number of the tools that are being adapted to the antimicrobial resistance surveillance of other pathogens.
Thank you. And one last question is, within a healthcare facility, so what are the basic steps that need to be done in Sentinel site labs before doing the sequencing in AMR, of AMR genes for bacteria in the reference lab? What are the basic procedures that should be done in the Sentinel labs is, I think, the essence of the question.
Yeah, whole genome sequencing or any reference lab service is so much more efficient if the Sentinel labs have really good identification. And so investing a lot in identification, we found that many of our labs are using, tube biochemicals, and sometimes they're cutting these down because of resource limitations. But you need enough biochemical tests to be sure that you're identifying your organisms, at least to the genus level, because this now tells you what should be referred to the reference lab. And so really boosting identification techniques to make sure that you're doing good identification, either by moving to a commercial system like API or Microbact or something, or by increasing your tube biochemicals so that you can get to genus level. The other thing that's really helpful for any reference lab service, including whole genome sequencing, is having good metadata. So the lab has to integrate the data that it generates with data from the hospital. So we know which ward is this from, what was the diagnosis, and so on. Because attaching that metadata to the whole genome sequence can be very, very powerful in providing the Sentinels with data that they can actually use to improve patient care, to improve infection control, to improve antimicrobial stewardship.
Thank you so much, Dr. Okeke, for providing answers to each of the questions. Please feel free to respond to some of the questions that might not have been responded to in the live Q&A session on Zoom. And thank you so much. With this, we go to the third session, and our third presentation is going to be presented by Dr. John Clemens. The presentation is on evaluating vaccine protection in clinical trials, efficacy versus effectiveness. Dr. Clemens is a senior scientific advisor at the IVI, and over to you, Dr. Clemens.
Hello, my name is John Clemens. I'm a senior scientific advisor at the IVI and an adjunct professor at the UCLA School of Public Health. And I will be talking about evaluating vaccine protection in clinical trials, efficacy versus effectiveness. So the gold standard designed for evaluating vaccine protection in a phase 3 trial is the individually randomized clinical trial design, in which a study population is assembled from a target population and then individually randomized by individual person or subject into experimental or comparison groups, which are then followed concurrently and longitudinally for the comparative occurrence of the target infection, which then forms the basis for calculating vaccine efficacy. Now, the goal of these phase 3 trials is to measure the full protection directly conferred upon a healthy vaccinated individual when the vaccine is administered in an appropriate fashion. And the question addressed is, can the vaccine reduce the occurrence of the target infection in an acceptably safe fashion under idealized condition? This is vaccine efficacy. What tactics are used in the trial design to ensure measurement of efficacy? Well, firstly, the target population is at high risk for infection and likely to respond to vaccination. We often will exclude subjects with significant comorbid conditions that reduce immune response, and these are subjects generally without pre-existing immunity, so that we would exclude people if they are previously infected or vaccinated. The vaccine formulation has the primary goal of maximizing immunogenicity. Practicality is secondary. Vaccine storage is done under optimal conditions. Administration follows rigid scheduling, and there's a high level of training and supervision of vaccinators to ensure correct administration. The control group would be persons who receive an agent without activity against the target infection. Generally, we would exclude individuals who are receiving other vaccines or drugs that might interfere with the immune response to the vaccine under study. The unit of allocation or randomization is determined by the goals of measuring only the direct effect of the vaccine in reducing susceptibility to the infection in the vaccinated person, as well as the goal of minimizing sample size requirements. And typically, this involves randomizing individuals rather than groups of individuals. Target outcomes in these trials are generally defined with restrictive biological criteria intended to demarcate outcomes that are most likely to respond to vaccine-induced immunity. The index of vaccine protection in these trials is vaccine efficacy, 1 minus the incidence of the outcome in vaccinees divided by that for non-vaccinees times 100%. This represents or measures rather the proportionate reduction of the target outcome attributable to vaccination, so that the magnitude of protective efficacy is not arithmetically affected by whether the incidence in non-vaccinees is high or low. And in analyzing protection, the group under analysis tends to be people who receive the complete regimen of the assigned vaccine or comparison agent per protocol. Outcomes are those infections that occur after a so-called immunogenic window, that is, after a period of time after completion of vaccination, when it can be presumed that the subjects have developed full immune responses. And the duration of follow-up is often the minimum interval required to obtain precise estimates of protection. Why do we do all these things? Well, one reason is to maximize the detectability of vaccine protection if it exists by reducing background noise and measuring the full effect of vaccination. The net result being reduction of sample size requirements and trial costs. The second goal for these tactics is to provide reproducible estimates of vaccine protection when the vaccine is tested in different trials and places. This would include tactics to remove the background noise and to measure the full effect of vaccination, the use of individual randomization to isolate our evaluation of protection from herd effects, that is to not measure herd effects, and use of the protective efficacy index, which is not arithmetically affected by how high the incidence of the outcome is in the control group. While having many strengths, this approach in phase 3 trials may leave uncertainties about vaccine protection after they are completed, indeed after the vaccine is licensed. We may have questions about the impact in a wide spectrum of recipients and under routine public health conditions as against ultimate outcomes such as death, as well as proximate outcomes such as infection, against soft outcomes such as disability, as well as hard outcomes such as biological infections, against long-term as well as short-term outcomes, and including herd, as well as direct vaccine protection. Because of these uncertainties, we typically see phase 4 studies of vaccine protection conducted after a vaccine is licensed and introduced. These studies measure protection based on the comparative occurrence of outcomes in persons who are or are not vaccinated in the course of routine practice after introduction. The common designs being cohort studies in which one begins with vaccinated versus non-vaccinated individuals, follows them forward in time for the comparative occurrence of the outcome, or case control studies in which one compares cases who have developed the target outcome versus controls those who have not for the antecedent receipt of vaccination. When cases and controls are assembled from a specific diagnostic test, with cases being test positive, controls being test negative, we call this a test negative design. An example of test negative designs was published in the New England Journal from Qatar recently, evaluating the Pfizer COVID-19 protection against different variants in Qatar, and comparing PCR positive persons versus PCR negative persons, and looking at antecedent rates of vaccination to determine protection against infection and disease according to various degrees of severity, various doses received, and various durations. These studies, these observational phase studies, have been very useful, but we have to be honest that there are limitations to these studies. There's the intrinsic problem of susceptibility to bias, in contrast to randomized double-blind trials, and the practical problem that the studies are often undertaken in an ad hoc rather than a systematic fashion, so that they are not systematically assessed by regulatory authorities as are randomized trials. As well, we have to realize that there are several prerequisites for conducting these observational studies. We need high quality information about vaccination status, about indications for vaccination, and other potential confounding variables for analytic adjustments. We need good medical infrastructure for making diagnoses and accurate records of these diagnoses. And if we're thinking about doing these studies in developing countries, fulfillment of these criteria is distinctly uncommon. Well, what can we do in these settings? Well, one approach would be to conduct vaccine effectiveness trials as distinct from vaccine efficacy trials, in which the question posed is what are the practical health outcomes, both beneficial or not, that are attributable to a vaccine when the vaccine is administered under the ordinary, not the idealized conditions of an efficacy trial, but the ordinary conditions of a public health program. How do we accomplish the goals of an effectiveness trial? Well, effectiveness trials are similar to efficacy trials in that we endeavor to maximize scientific validity with suitable eligibility criteria, randomized allocation of groups of subjects to vaccinated and control groups, and unbiased ascertainment of endpoints, as well as to preserve ethical propriety. But it's different. They are different from efficacy trials in that they endeavor to maximize the applicability of the results to public health practice and cast the results in terms that are of greatest use to public health policy makers. What tactics are used in effectiveness trials? Well, in an effectiveness trial, the target population is all persons who realistically might receive the vaccine in a public health program. The vaccine formulation is one with practicality being as important as immunogenicity. Vaccine storage and administration is done under the ordinary conditions and schedule of a public health program. The control group is defined by the public health question. It may be an inactive alternative, such as an efficacy trial, but it may be an active alternative if that's the public health question. There are no restrictions on other vaccines and drugs given if these constitute routine practice, and the unit of allocation is demarcated to measure relevant public health effects of vaccination, including both direct effects to individual vaccinees and herd effects. To do this, the unit is usually a geographically defined cluster of individuals rather than individuals per se. So these trials are generally cluster randomized trials. To assess protection in an effectiveness trial, the target outcomes are less restrictively defined than in efficacy trials. We study both soft and hard outcomes, ultimate as well as proximate outcomes, and long-term as well as short outcomes. The index of protection for an effectiveness trial may include the ratio of protective effectiveness analogous to an efficacy trial, but often will be the rate difference, the difference in the occurrence of the outcome in non-vaccinees versus vaccinees, which would estimate the absolute decline of the incidence of the target outcome attributable to vaccination. This directly addresses such pragmatic questions, such as if 1,000 persons are vaccinated, how many illnesses will be prevented? And for analysis of protection, the group under analysis would be all persons targeted for participation in the public health program, whether or not they actually participated. Outcomes are those occurring from the time of initiation of the vaccination program rather than after an immunogenic window, and we endeavor to measure herd as well as direct protection. So the basic architecture of a cluster randomized effectiveness trial looks identical at a high level to that for an individually randomized trial, except the unit of randomization at R is the group cluster, the cluster or group rather than individuals per se. How do we analyze cluster randomized trials? Well, this shows two representative clusters from a cluster randomized trial comparing intervention clusters receiving a study vaccine versus control clusters receiving a control agent. You'll see that in each of these clusters, there are people who actually get the assigned agent, be it a study vaccine or a control agent, and people who don't. And there are associated ARs, attack rates of the outcome, in each of these four subsets. To measure indirect vaccine protection, we contrast the attack rates in non-recipients of the control agent versus non-recipients of the study vaccine to estimate total protection, which may be thought of as a combination of direct protection and indirect effects benefiting vaccinees. We can contrast those rates for study vaccinees versus recipients of the control agent. And for overall protection, the overall difference in the occurrence of the outcome, regardless of what people received in the clusters, we simply contrast those overall rates in these two types of clusters amongst everybody in the clusters, regardless of what they received. You can see that we can estimate vaccine direct protection by contrasting the attack rate in study vaccinees versus non-vaccinees in the intervention clusters. However, in contrast to these other measures, which I've just mentioned, these are not randomized comparisons. An example of a cluster randomized effectiveness trial was conducted by the IVI years ago in Calcutta, India, where diseases like typhoid fever thrive. In one such slum in Calcutta, 80 geographic clusters were demarcated as shown and randomized to receive either VI polysaccharide typhoid vaccine or hepatitis A vaccine control agent. Individuals aged 2 and over in these clusters were eligible to receive the particular vaccine assigned to the cluster. This slide shows the analysis of total vaccine protection among VI vaccinees versus hepatitis A vaccinees, 96 versus 34 episodes of typhoid for 65%. This slide shows the analysis of indirect protection in this cluster randomized trial, comparing typhoid incidence in non-vaccinees in the hepatitis A clusters versus non-vaccinees in the VI clusters, 31 versus 16 episodes, 45% statistically significant indirect protection of non-vaccinees. And this is the analysis of overall protection of all residents in the Hep A clusters versus all residents of the VI polysaccharide clusters, regardless of what was received, regardless of age, 127 versus 50 episodes for 60%. Now, this may seem like a not terribly impressive level of overall protection, but if you recognize that the level of coverage of the VI clusters with VI vaccine was only about 60%, you come to the conclusion that the combination of direct and herd effects resulted in overall protection that would be equivalent to a vaccine whose direct protection was 100%. So what might be the potential roles of efficacy and effectiveness trials? I would see them as complementary, with efficacy trials serving as screening tests of vaccine performance, providing the basis for licensure, which will usually be needed before conducting effectiveness trials, with effectiveness trials then as confirmatory tests of vaccine utility for vaccines found to be promising in efficacy trials, but doubt remains about their practical usefulness. They might be termed 3B trials in this respect. Effectiveness trials may be of particular importance in less developed settings where there are extremely limited financial resources for inclusion of new vaccines in public health programs, so that a practical cost effectiveness and measurement of effectiveness is of great importance before decisions to introduce vaccines, and in which, of course, as I mentioned, there may be difficulties in conducting observational phase 4 studies. So in summary, in current practice, individually randomized trials of vaccine protection usually measure vaccine efficacy, and these efficacy trials constitute pivotal studies for licensure. The efficacy perspective has the advantage of maximizing the detectability of vaccine protection, thus reducing sample size requirements, and of increasing the reproducibility of results in different settings. However, it does not necessarily provide information that is needed to make public health policy about the introduction and use of these vaccines. Studies of vaccine effectiveness, which evaluate vaccines as they're used in practice, are typically conducted after licensure. These post-licensure evaluations of effectiveness are usually conducted in an ad hoc fashion using observational or non-experimental study designs. They are susceptible to bias and rely on data that are not routinely available in less developed settings. The effectiveness trial paradigm may overcome several of these limitations of the observational post-licensure studies, especially relevant for developing country settings. Thank you very much.
Thank you very much, Dr. Clemens, for the very nice and informative presentation. We have a couple of questions from the audience. I will read them out for you. The first question is, COVID-19 vaccines are being used under emergency use authorization. And in this context, what would be the challenge of conducting phase 4 trials? Did you hear me?
Thanks for that question. And the COVID pandemic presents very unusual challenges and timelines for vaccine evaluation. And frankly speaking, at this point in time, with the urgency of vaccine rollout globally, I think it becomes ethically very problematic to consider randomized effectiveness trials. And as time wears on, it will become more and more problematic. I think that the effectiveness randomized trial paradigm really is more applicable to the normal circumstance of normal vaccines against normal endemic diseases in which there is not global urgency.
Thank you. The next question is, given the when and waxing of the incidence over time, how can we best control this when we utilize cohort studies in evaluating efficacy? Would it be using by doing a longer study period or using specific cutoffs based on the peak incidence or prevalence?
Well, I think we need to distinguish between vaccine protection and how long vaccine protection occurs. They really are separate questions in view of the tendency for vaccine-induced immunity to wane over time. A common misunderstanding in designing trials is simply to extend duration of surveillance to increase cumulative incidence and meet sample size and statistical power requirements. But the question then differs. The question of short-term efficacy versus long-term efficacy or protection, those are different questions.
Thank you. And the third question is, with many infections in COVID-19 being asymptomatic, it's quoted as greater than 85% in the question, how would this affect vaccine efficacy evaluation? And would it be relevant to actually evaluate asymptomatic infections as measures of efficacy for vaccines, for COVID-19 vaccines specifically?
So the answer is it depends. from a public health point of view right now, the major public health concern is reduction of severe illness and death. And that then is the logical target for evaluation. However, in the longer term, control of COVID by vaccine and non-vaccine interventions will require reduction of transmission of infection, not merely reduction of severity of infection among vaccinees due to the vaccine received. And as time progresses, issues of what are called vaccine impact, that is the overall impact of a vaccine on a population, considering the benefit both to vaccinees and non-vaccinees, will become of greater importance, and correspondingly, evaluation of effects on transmission and potentially asymptomatic infection will also be more important.
Thank you. And the next question is, it's becoming quite possible that regulatory agencies around the world may approve second and third waves of COVID-19 vaccines based on non-inferiority immunogenicity data if immune correlate of protection gets established, but may enforce companies to conduct real-world effectiveness studies. In the context of COVID-19, what would be the appropriate study design? Should a test negative design be preferred, similar to flu studies? What are the variables? What variables should investigators consider collecting efforts to minimize selection bias?
Right. Well, the proper studies to be done will be the studies demanded by the regulatory agencies. Clearly, the test negative design has emerged as the darling of non-randomized evaluations of vaccine protection, in part because they're very easy to do, in part because when done well, they confer more protection against bias than other observational designs. But in the last analysis, it's going to be up to the regulatory agency's prescription.
Thank you. And another question is about sample size calculation for vaccine effectiveness studies. What are the standards that should be followed in vaccine effectiveness for sample size calculation and vaccine effectiveness studies evaluation? And does this vary during a pandemic setting, is a question.
So the calculation of a sample size in an effectiveness trial follows the same logic and in general the same formulae as for an individually randomized trial, except that it must account for the fact that you're randomizing clusters of individuals rather than individuals per se, which introduces what statisticians call a design effect. In simple terms, an intercorrelation of the probability of people in the same cluster having the same outcome. That inflates the sample size requirements for cluster randomized trials over individually randomized trials.
Thank you, Dr. Clemens, for addressing all the questions, and please feel free to answer to some of the questions that were not answered in the live Zoom Q&A session. Thanks so much. And with this, we move to the next presentation, the fourth presentation. Our fourth presenter is Dr. Mark Jeet from the London School of Hygiene and Tropical Medicine. Professor Jeet is a vaccine epidemiologist at the London School of Hygiene and Tropical Medicine and a visiting professor at the School of Public Health, University of Hong Kong. His research group focuses on epidemiological and economic modeling of vaccines to support evidence-based public health decision-making. And Dr. Mark has published over 250 papers covering a range of vaccine-preventable and potentially vaccine-preventable diseases, including COVID-19, measles, HPV, pneumococcus, RSV, dengue, and group B streptococcus. As well as methodological papers advancing ways vaccines are evaluated, have been covered by his papers. This work has influenced many of the major changes to immunization policy in the UK and globally, and he also organizes or contributes to academic and professional courses on vaccine modeling, economics, decision science around the world. And for today's session, Dr. Jeet's presentation will be on the role of health economics and financing on new vaccine introduction. And he will be joining us live on Zoom for this session. Over to you, Dr. Jeet.
Great. Thank you very much for the introduction. I'm delighted to be able to be here at the IVI vaccine course again after a one-year hiatus. And I must say that this looks like the best attended vaccine course I've ever been to. I've never spoken at a vaccine course with 2.1 thousand people. So wherever you are in the world, it's great to be speaking to you. I'm going to speak about the role of health economics and financing. So all the economic and financial aspects of new vaccine introduction. Maybe we could start with the next slide. So the topics that I'm going to cover in this talk include three items, really. First of all, we want to understand what's the role of health economics when we're looking to introduce a new vaccine. Why might we want to consider economic aspects in this? Second of all, we want to know what are the basic tools that we can use in order to plan financially and economically to see what are the financial requirements and what's the value for money of a new vaccine before vaccine introduction. And third of all, what I'm going to talk about is true of many health economic evaluations, but what I'm going to ask, are there any special features of vaccine programs that we need to take into account when conducting an economic analysis? So let me start, if we can move to the first slide. Let me start by asking the motivation for using health economics. Why are we using health economics to consider new vaccine introduction? I mean, at this point, if we were in a room, I'd ask people, how many of you have actually studied economics before at any point of your education, whether, in high school, in university, as a, undergraduate course? But I mean, I'm not going to ask that to 2,000 people. But what I want to start off is some very basic economic concepts, which some of you might have heard before. The most basic, I think, is the idea of scarcity. This is what has been called the fundamental problem of economics, is the problem of scarcity. And the problem of scarcity is that many of the things that we want, that we might demand, that we might need, we have insufficient supplies of. And so we have to decide who gets the supplies, right? So economics is interested in the goods, the resources, the objects, where the supply is not enough to meet demand. And therefore we say that that resource is scarce. And well, most resources that contribute to healthcare are scarce. So very easy to think about examples in healthcare. So you think about the resources that go into healthcare: money, staff time, hospital beds, drugs, whatever it is. And these resources are always limited and insufficient to meet all the demands for healthcare. Now, the second key concept to look at is the idea of an opportunity cost. When something is scarce, when the supply of something is not enough to meet the demand, then when we choose to allocate that supply to meet a certain kind of demand, we lose the opportunity to use it for something else instead. And the next best alternative use of that resource is called the opportunity cost. For instance, you might have a healthcare budget and you might decide, we're going to use this budget to buy a vaccine this year. We're going to pay for the COVID vaccine, say. That means you can't use the same amount of money to do something else, to build a new hospital or build a new school or road. And whatever's the best alternative use of the money is what you're giving up. It's called the opportunity cost of the choice. Or for instance, you can say, well, all of you decided to come, 2.1 thousand of you decided to come for this course this morning, afternoon, evening, depending where you are. Instead of doing something else, instead of doing something else in your work, instead of meeting your friends, instead of, cooking a meal for the family or whatever it is. So whatever's the next best use of your time, instead of attending this course, is the opportunity cost, but hopefully the value of this course is worth it. So let's move on to the next slide. Since choices in healthcare involve opportunity costs, since we have limited resources and we have to decide how to allocate it, therefore, we need a mechanism to decide how we're going to allocate these scarce resources. And the mechanism we use to allocate is called our mechanism of rationing. Or nowadays, people like to use the word priority setting instead of rationing, but it basically means the same thing. It means that we have to choose to use these resources for something X, and therefore we can't use it for something Y. So we might decide we don't want to have a explicit system for rationing. We don't like that idea. So still, rationing decisions have to be made, but nobody actually understands. Nobody can see how they are made. So allocation is still limited by supply, but the criteria by which these decisions are made are not made explicit. They're made by a committee who doesn't say how they made the decisions, an implicit system of rationing. That's the system that was actually used in the country I'm from the UK, up to about the mid-1990s. And it was what was called the Postcode Lottery. Why was it called a Postcode Lottery? Because often what would happen is that some healthcare authorities in a local area would decide, we're going to supply this drug. If you get cancer, say, you're going to have this drug. But you might live just a few streets away, and the next local authority might decide we're not going to pay for this drug. So people said it was a bit of a lottery. It depends on where your postcode is. If you live there, you don't get the drug. If you live there, you get the drug. And there was a lot of unhappiness about this postcode lottery. And that led to the creation of an agency called NICE, the National Institute for Health and Care Excellence, which would make these decisions on behalf of the whole nation and decide actually based on explicit criteria which drugs should be funded by the health service. So what are other ways to allocate healthcare? A second way is you could say, well, we want to allocate healthcare the way we allocate many other resources, and that's by price. So if people are willing to pay what it costs to produce it, if there's a willing seller who's willing to supply the healthcare for a certain price and a willing buyer prepared to pay the money for that healthcare, then the healthcare transaction can take place. That's the market economy. That's how many things are allocated. If you want to buy a mobile phone, then are you willing to pay the price that Apple or Samsung charges for the phone? If you are, you get the phone. If you don't, you don't, right? Now, in healthcare, interestingly enough, price-based allocation mechanisms are much less widely used compared to most resources in society. Every country in the world, no country in the world purely allocates healthcare based on price, although there is a role for price-based mechanisms in many, although not all countries. Why is that? That's for a number of reasons, but I think one of the most important is what I think a value judgment, a moral reason, which is that many, if not most people, feel that it's unethical to deprive someone of healthcare because of their inability to pay, when you could actually save their life if they could say someone has pneumonia, you could give them an antibiotic, but they can't pay for it. We have a moral obligation to save their life if it's possible. So as a result, there are many non-market mechanisms used to allocate healthcare. I mean, there are other reasons, what economists call market failure in the healthcare market, which require government intervention, but I won't go into those right now. So if you don't want to allocate by price, another means you could allocate is to say, let's allocate by waiting lists. Let's say that people's willingness and ability to wait should determine the allocation. So they first come, first serve, basically. If you come and there's already all the healthcare is allocated, then you have to wait until more healthcare becomes available. So allocation by waiting list, ability to wait. Now, a fourth way to allocate, because you might say, well, that's not always the best thing to do. What if someone just had a stroke and they're being rushed to hospital? Are you really going to wait until those 10 people in front of them are treated before you treat that person with a stroke, when you could save their life by prioritizing them, right? So a fourth way to allocate is evaluation of needs, a needs-based allocation mechanism. What does that mean? That means that we choose a criteria by which we allocate healthcare. It might be to maximize the health of the population, or it might to maximize the equity of health. We want to, first of all, allocate to meet the needs of the people with the worst health, that we can help the most. Whatever your criteria is, you then choose your allocation to maximize this criteria. And this method, as you can see, really sounds like the most evidence-based and fair way of allocating healthcare resources. The disadvantage is this is also the most complicated because you need to do this evaluation, and then you need to decide how to allocate according to your evaluation. So there's quite a lot of technical complexity in this allocation. So in the rest of this talk, I'm going to talk about some of the challenges and solutions for the technical complexities. But first of all, let me ask, when it comes to vaccines, what really is the decision about allocating healthcare resources? Should we allocate resources for healthcare? Well, I've talked a lot about the UK. Let's look at a country. Let's look at some settings which are more resource limited. So if we go on to the next slide, let's do a thought experiment. Let's say, for instance, you are the Minister of Public Health of the Democratic Republic of Congo. Really important job because your budget will be quite limited. Your budget in 2015 was $17 per person. And so you have a great deal of healthcare needs, and you need to use this limited budget in the best way possible to maximize the health of your population. How would you do that? Well, one thing that many countries do, if we go on to the next slide, is, well, go to the WHO or go to the World Bank and ask them, what do you suggest? And in fact, this is decades old, but this is still very, very relevant today. The World Bank actually funded an analysis that looked at a whole package of different interventions that could be introduced into a healthcare system. And they concluded that there was a package of interventions which really all countries should consider because they are very, very good value for money. They cost very little to save a year of life, to prevent a DALI. A DALI is really a unit of life free of disability. And one of the most value for money items is what they call the EPI Plus program. What's the EPI Plus? That consists of the EPI vaccines, the very basic schedule of vaccines that WHO recommends all countries introduce, plus vitamin A supplementation for children. So the conclusion was the EPI Plus package is one of the best uses of a healthcare budget. So vaccines are in general very, very good value for money. Now, there's a problem though. This is the EPI series of vaccines, the vaccines that countries have been using since the 1970s. What about today? And if we can go to the next slide. Well, the problem is that the number of vaccines, the opportunity, but also the challenges that the number of vaccines in different countries has grown dramatically since the 1970s. Many new vaccines have been developed and licensed since the 1970s. So if we go on to the next slide, there's another thing that's very important about these vaccines. That's the nature of these vaccines have also changed. So the EPI vaccines, the vaccines that were evaluated by the World Bank, these are extremely cheap. Measles-containing vaccine might cost less than $1 a person, for instance. They're against common infections. If we didn't have vaccines against measles or against rubella or against diphtheria, many millions of people around the world, almost everyone would probably be infected by these infections at some point. And they can all be delivered to infants or very young children. So every country in the world knows how to deliver EPI vaccines. Today, however, we have all these new vaccines like PCV, rotavirus. We've talked about dengue recently, HPV, and now COVID, which are, first of all, more expensive. Second of all, some of them are against less common infections, like some of the meningococcal vaccines, for instance, still against very severe diseases, but much less common than measles, for instance. Some of them suffer from partial or waning efficacy, so the effectiveness of these vaccines in the population is not as clear as measles-containing vaccines, which have over 95% efficacy with 2 doses, for instance. And often we're now trying to deliver them to non-traditional risk groups, not just infants or young children, but for instance, with the COVID vaccine, an entire population, with HPV to adolescents. So we need new methods of delivering these vaccines. And when we go on to the next slide, the consequence of this is that the cost of delivering and purchasing these vaccines has expanded dramatically. MSF estimated, for instance, in 2001, the entire series of vaccines recommended by the WHO cost only about 67 cents for the whole course. But by 2014, the price of this had increased more than 60-fold to more than $45 US. I mean, the big increases were the introduction, first of all, of Hib vaccine, followed by PCV, followed by HPV vaccines. These are life-saving vaccines, but they cost so much more than the EPI schedule. So how do countries decide which of these vaccines to introduce? And also, I guess, also very important, if we go to the next slide, the other big elephant in the room, really important vaccine, but really expensive also, is the COVID-19 vaccine, which costs anything from between, a few dollars a dose, still more expensive than the EPI vaccines, to maybe tens of dollars a dose. And UNDP estimates that actually low-income countries would need to spend more than half the entire healthcare budget in order to vaccinate the majority of the population against COVID-19. So we go on to the next slide. How do we decide then, how we're whether we should introduce these vaccines? Well, first of all, we have to decide what is the economic tool, what is the economic question we need to answer, and then we have to decide which tool to use to answer it. If it's a question of how much it costs, what we need is an affordability tool, an affordability study. This vaccine will cost so much per person vaccinated. Let's say it's HPV per vaccinated girl, for instance. Or if we want to ask, well, is it sustainable? How much are we going to pay for it year on year? Then what we need is a budget impact analysis. This is how much it will cost the entire budget over the next 10 years. If we want to know what's the benefit of this vaccine, how many deaths is it going to prevent, then what we want is an epidemiological model to project the impact of the vaccine. And if we want to bring the costs and the impact together, then what we want is a full economic evaluation, like a cost effectiveness evaluation, to look at the cost per DALY averted or cost per QALY gained of this vaccine. And finally, we can then turn that into a threshold price analysis, which says how much are we going to pay in order to buy, what is the maximum we should pay for this vaccine, which can also aid in price negotiations with manufacturers or with other external agencies. So the first thing I'm going to talk about is affordability. How do we know whether we can afford this vaccine, and how do we finance this vaccine? Well, Gavi has a useful chart, I think, which looks at a country when they start off, if we go on to the next slide, with quite a low budget for health, with a country with very, very limited resources. And then as that country gradually transitions to middle income status, gets a larger budget, the economy grows, then actually the amount that Gavi finances will decrease. So Gavi finances most of the cost of vaccine purchase for the poorest, most fragile economies in the world. And then gradually as they transition, the country finances a larger and larger share until the time they become fully self-financing. Then often they're guaranteed still a low Gavi price for a number of years, but they take on the financing themselves 100%. So when that happens, what are the other sources of financing? And that, maybe go on to the next slide, please. Well, you can look at both internal sources of financing from the country itself, but also external sources. So the internal sources can be internal revenues, the healthcare budget, the wider, general budget. Or it could be specially earmarked taxes, for instance, the so-called sin taxes against things like alcohol, tobacco, maybe sugar, because countries recognize that consumption of these items increases healthcare expenditure, and therefore the revenue from the taxes of these things go to financing healthcare. There can be internal sources where users pay, for instance, private health insurance or user co-pays. All of these can be very inequitable because they disadvantage the poorest people. Or there can be national trust funds where, for instance, oil-based economies put aside money to fund healthcare, or revolving funds where countries group together, like countries in the Americas. Then there can also be external sources of funding. I talked about Gavi support. There are also multilateral and bilateral instruments, the World Bank, ADB, the COVAX initiative for COVID vaccines I'm going to talk about in a moment. countries, debtor countries can offer debt relief so that debty countries can fund healthcare. Some countries have availability to raise funds on the capital markets to get loans, to issue bonds, but not all countries have the credit rating to do that. But there can be more innovative instruments like AMC or IFFIM mechanisms by which maybe countries can pay in to fund other countries accessing lower prices through some of these mechanisms. So we go on to the next slide. One challenge countries have at the moment is really deciding how to finance COVID-19 vaccines. And here there's a whole range of procurement options, ranging from waiting for a bilateral donation from a country that can afford more vaccines to the COVAX initiative, which has different sections of it. One section is the AMC mechanism that's open to the 92 poorest countries in the world, or moving on to self-financing mechanisms, where there are two different arrangements, one with a higher price, but which offers greater flexibility. Moving on to bilateral deals, direct deals with manufacturers, and then finally, establishing local manufacturing capacity. So as we go down this list, it offers more and more flexibility. The most flexible is to build your own manufacturing capacity in your country. And then you're not at the mercy of whatever supply and price is available overseas. But it also requires the greatest time and the greatest resources and technology in order to do this. The lowest cost is to wait for a donation, but then there'll be no flexibility or guarantees about the type of vaccine a country gets or the timing of delivery. And then COVAX is an attempt to give countries a bit more flexibility. COVAX aims to deliver a certain percentage of doses to all countries in the world. But at the same time, there are limitations because COVAX only has a limited supply of vaccines itself. So again, there are different options for COVID-19 vaccines. And if we go on to the next slide, after you've decided, well, we can afford this vaccine, it doesn't mean you should buy it straight away. You also need to consider the value for money, the cost effectiveness of a vaccine. And so, as I said before, you need to compare both the costs of the vaccine with the consequences of the vaccine. And really, you need to consider two worlds and compare them to each other. You need to look at a world without the vaccine and say, how much does it cost to treat this illness? And what health burden, how much suffering is imposed if people get sick? And compare that to the world with the vaccine. So you say your burden of illness decreases, but now you have to pay to buy the vaccine, and then your health burden decreases. And then if we go to the next slide, we compare these two worlds. We take we really look at this fraction and we say, what's the incremental cost? What's the difference in costs we pay when we introduce a vaccine? And then we divide it by the health impact. What's the difference? What's a health burden that we avert when we introduce this vaccine? And then you come up with a ratio like a cost per DALY averted or a cost per QALY gain. DALYs or QALYs are generic measures of health, which basically represent 1 year of life in good health. And so we ask how much they cost to buy this year. And if the cost is low enough, then you say, actually, this is good value for money. But if we go to the next slide, when it comes to vaccines, unlike many other health interventions, we need to consider special considerations. We need to consider what's happening between the pathogen and the host, for instance, what's happening in the population as this infection can spread. And so when we vaccinate people, we're not only benefiting the vaccinee, we're also preventing onward spread of the vaccine. But we also have to consider ecological effects like maybe serotype replacement for PCV, which might decrease the cost effectiveness, but also herd immunity, which might increase the cost effectiveness. We might consider that the time horizon we should look at, the length of time we follow the impact of a vaccine might be very long. Like for HPV vaccine, we need to look decades in the future to get the full benefits, unlike a drug, for instance, which probably has the benefit immediately. And we need to consider the economic scope. And if we go to the next slide, I think we have an example that's been with us for the past 2 years really prominently, and that's COVID-19. And really, here's an analysis by the RAND Corporation, for instance, really showing that the economic benefit of the COVID vaccine could be measured in hundreds of billions of dollars globally, especially if the vaccine can be delivered not only to the so-called vaccine nations, the countries with their own vaccine manufacturing like Europe, USA, China, India, Russia, but also the countries which don't have vaccine manufacturing themselves. If the vaccines can be rolled out sufficiently, then the world can really save up to $1 trillion US from these vaccines. So if we can go on to the next slide, I'm going to sum up now with some key messages. First of all, I've talked about the way we allocate resources in healthcare and saying health economic evaluations is the way we can use to ensure that these decisions are explicit, evidence-based, and needs-based. Second of all, new vaccine introduction, we need to consider how much does it cost? Can we afford it? Where are we going to get the financing from? And third of all, is it good value for money? What impact are we getting, including these ecological effects? And third of all, we need an economic evaluation framework, like a cost effectiveness analysis. This is a somewhat technical process, but this allows us to consider all these different aspects together and decide whether or not to introduce this vaccine. So that's all I have today. If we go on to the next slide, it's a bibliography if you want to do some further reading, but I'm really happy to answer any questions.
Thank you so much, Dr. Ajit, for the very nice presentation. We have some questions from the audience, and I will forward them to you in reading. So the first question is, I understand that to be financed and reimbursed under the National Health Scheme, it has to undergo a health technology assessment. As different countries have different criteria to consider, such as budget impact and ICER, what do you think should be the biggest priority in evaluating novel vaccine technologies?
I think the advantage of a HTA, a health technology assessment, is it's really interdisciplinary. So you consider many aspects, and I think all these aspects are really important. So definitely you want to consider, what's called the pharmacovigilance criterion. Is the drug or the vaccine, is it safe? If it's efficacious, is the quality of the product good? If it can't pass these criteria, you shouldn't be using the vaccine, right? But the vaccines which WHO recommend, for instance, the pre-qualified vaccines have all passed these tests. But then you need to consider what's the local burden of disease. You need to consider the economic criteria. It might be a great vaccine, but if the budget is too small to afford it, you don't want to be stopping your measles vaccine or your nutritional supplements in order to introduce this new vaccine, right? You must make sure it's good value for money. And then I would say the wider criteria, is it acceptable? Is it the ethical considerations addressed? Those are also important. So I'd say you really need to bring all these different perspectives to the room, and that's the value of the HTA process.
Thank you.
Thanks for the question.
The other question we have is, in the context of COVID-19, from a health economics perspective, what needs to be done to realize vaccine equity, and in particular, convincing rich countries to release excess vaccines, for example, which are in their possession? Yeah.
I think that's such an important question. That is the big question about COVID vaccines for today, because we have a situation where, many countries in Africa have only vaccinated under 5% of the population, but some rich countries are already considering, giving a third dose to their population. And so I think there are many strong reasons for equity besides just justice, but also for vaccine equity. Some of the other reasons are so that the whole world can go through an economic recovery, and that benefits everyone. I think another reason is actually to prevent new variants from constantly emerging. I mean, many people have said, the COVID pandemic will not be over for anyone until it's over for everyone. So as long as there are countries with, high burden of disease, with continued high levels of circulation, we're going to keep on having emergence of new variants, and we keep on having to update our vaccines. So the only way to emerge from this is to make sure that all countries have high levels of coverage of vaccines. So I think some of these arguments are really important to be made to ensure good vaccine equity for COVID vaccines.
Thank you, Dr. Ajit. Another question is about government allocation of funds, basically. What's the great challenge or barrier to government allocation of sufficient budget to healthcare, despite evidence, for example, that vaccines, what vaccines can do in the long term? For example, fewer diseases, save time attending healthcare needs. Would this be related to, for example, issues of budget impact analysis? That's the question.
Well, I think one challenge is that many countries have a very small budget for healthcare in general, not just vaccines. And so often the value of these economic tools is that they are needed in order to make a case from the Ministry of Health to the Ministry of Finance, because the Ministry of Finance may have to consider not just funding healthcare, but funding many other good uses of money, build a new school, build a new road. And therefore, they need to see the economic case for funding a vaccine. I mean, the Ministry of Health may say it's going to save many thousands of lives, but the Ministry of Finance says, well, but what is the economic case? Well, what value will this bring to the country? Why should I do this instead of building a new airport or a new school, right? So I think the value of economic evaluations is it helps that case to be made to the Ministry of Finance so that they can compare, well, if I spend $1 million on this vaccine versus $1 million on this road, how can I compare them to each other and decide, okay, I'm going to increase the healthcare budget because I can see that this is a good investment.
Thank you. And the last question is, what's the reason for a high price of vaccines today? Is it primarily patents, monopoly rights on vaccines, or does it have to do with pharmaceutical companies to make unprecedented profits, for example? Yeah.
There are many reasons, so it's not completely straightforward. I mean, one reason is that genuinely the cost of producing these vaccines sometimes is higher because some of these vaccines use complex technologies like maybe mRNA technology for some COVID vaccines or quite complex adjuvants for some of the more recent vaccines. Another reason is really the economic model that many vaccine manufacturers use, in which what the economic model is really that they will, invest in many different vaccines or drugs, and they expect, maybe 90% of them to fail, but the ones that succeed are supposed to pay for all the ones that fail as well. And so when they first introduce the vaccine, then actually they charge a very, very high price in order to make back their R&D costs, including for all the failed vaccines or drugs. I think in order to bring the price down in the long term, in the short term, we really need mechanisms like Gavi, but in the long term, what we need is really more entrance to the market. So we need, many manufacturers around the world, including manufacturers that seem to be able to produce vaccines at scale at low prices, like for instance, Serum Institute in India, to also enter the market. And then we see the price of the vaccines decrease, like we've started to see for things like rotavirus or HPV, for instance, as their new entrance into the market.
Thank you so much for answering to all to most of the questions. And please feel free to also provide some response in the Zoom Q&A platform if there are any unanswered questions. And thank you.
Great, thank you. Glad to be here.
And with this, we move to our last presentation, the fifth presentation, which is on Introduction to Disease Transmission Modeling by Dr. JongHoon Kim, who is a research scientist at the International Vaccine Institute. And Dr. Kim, please.
Hi, my name is JongHoon Kim. I'm an infectious disease epidemiologist who works on infection transmission modeling. Diseases that I've studied include HIV, polio, and recently COVID-19. And today I'm going to talk about the introduction to disease transmission modeling. So today my title is Introduction to Disease Transmission Modeling. This slide shows an overview of the talk. I'm going to start with an example of how models can inform COVID-19 mitigation policy and what disease transmission models can do. We can learn from these examples. And then disease transmission models and built on the mechanism of how transmission spreads. So we're going to study transmission dynamics during an outbreak. Once we have become familiar with transmission dynamics during an outbreak, then we are in a position to learn about important concepts in infectious disease epidemiology, such as basic reproduction number, herd immunity, or critical or minimum, that's vaccination threshold. Once we learn those concepts, then we move on to actually see how the model can be implemented. And then for real world questions, simple models can provide sufficient answer. So we're gonna look at how simple models can be extended to provide a better answer to real world questions. Examples of those extensions include age-dependent mixing, asymptomatic transmission, or special movement ensemble model, all based on COVID-19 examples. So I'm going to start with a news article that says policymakers are trying to make use of model-informed decisions. So US and UK are trying to make decisions based on a research article, which turns out to be developed by researchers at Imperial College London in 2020. This article shows a likely outcome of an epidemic without interventions for US and UK. It shows a rather depressing outcome, about 2 million deaths in the US and 500,000 deaths in the UK. So if you look into the article and more, it not only provides a likely outcome of an epidemic, but also it provides a potential impact of intervention strategies, in this case, such as case isolation, household quarantine, or general social distancing, or school and university closure. So you can, these intervention measures may delay an outbreak or even decrease an outbreak size. So in summary, the models that we're going to talk about today will help project how infectious disease will progress to show a likely outcome of epidemic. The variables interest, it can present variables we are interested in, such as COVID-19 cases, death, or demand for ICU beds or ventilators. But also it can show an impact of potential intervention strategies such that policymakers can be informed. So far, we haven't defined what model is, so I think it's safe to make a definition to let you know the definition of modeling. There are many kinds of modeling, but we are interested in scientific modeling, which can be defined as the generation of physical, conceptual, or mathematical representation of real phenomenon that's difficult to observe directly. So as you can imagine, we're not going to be able to develop any real physical thing, but we'll be talking about mathematical model, but the mathematical model usually starts with a conceptual model. So we're gonna discuss conceptual or mathematical models. In epidemiology, there's mathematical models implemented or practiced in two main kinds. One is just statistical, and the other is a mechanistic model. Maybe based on if you're an MD or an epidemiologist, you are more familiar with the statistical models. Statistical models is rather simplistically defined is fitting a line or curve to data and make extrapolation out of that. So it doesn't usually account for how transmission occurs if we are dealing with the transmission infectious disease data, so that it's not well suited for long-term projections. One example would be just fitting a parametric, for example, normal curve to a frequency data, in this case, a serial interval. Just in case you don't know the serial interval, which is that serial intervals is defined as the interval between clinical onsets in a successive transmission chain. So if you think about the serial interval data generated, it can be rather complicated. It can be, for example, at least 3 different scenarios can be formed. For example, a symptomatic transmission. You need at least 2 people, the primary and secondary case, and transmission happens once the primary case, the circle represents illness onset. Primary case develops symptoms and transmission happens. So you have a positive serial interval. And the second scenario possible for COVID-19 is pre-symptomatic transmission. So primary case develops symptoms, but then transmission happens before the primary case develops symptoms. But then there are considerable similar incubation periods, so you still have positive serial interval. The last case will be pre-symptomatic transmission happened before the primary develops symptoms. And in this case, in addition, the secondary case experiences a shorter incubation period so that you have a serial interval is the shorter on negative. So then you can, all this the simple frequencies curve, it might be relatively complex mechanisms that are running behind. So mechanistic model is trying to accommodate or account for those transmission mechanisms. It has many different names. Some people call it an epidemiology model, or transmission model, or dynamic transmission model, or dynamic model, mathematical model, or some people call it dynamic compartment model. One of the more common things for in relation to COVID-19 is SEIR. So you define 4 different classes for a population. So there are susceptible subpopulation, exposed, infectious, and recovered subpopulations. Since we include mechanisms, it can be possible to rather project in a long-term future. But of course, there's no guarantee that our mechanisms will hold in the future. So one example would be, think about, this one will be one example of a conceptual diagram or a conceptual model. So you divide into population into many different classes. Here, susceptible, exposed, here, presymptomatic, asymptomatic, infectious, recovered, and hospitalized. And then you implement some mechanisms how asymptomatic or symptomatic, presymptomatic transmissions, we're talking about orange line here, generates infection, meaning influenza susceptibles become susceptible. So you define some sort of mechanisms how S becomes E becomes P, and things like that. So this is one example. We're going to discuss that more in detail later. So key is to incorporate some sort of realistic mechanisms how the disease spreads in the population. So that's the key. So we're going to look at what kind of mechanisms are going on in an outbreak. This is showing the daily confirmed COVID-19 cases. As you can imagine, there are multiple factors influencing the shape of this trajectory of daily confirmed cases. We're not going to be able to explain all this, but rather focus on a small subset of the data, maybe just one outbreak in a certain area. Even then, this one is rather complicated. We are more resort to the simplistic setting, such as this model projection. You have a rather smooth shape outbreak, but this is, maybe you could say this is not realistic, but in reality, we can also see something similar, very similar outbreak, very rapid growth and then rapid decay. If the outbreak happens in a short period of time with no major intervention. So we're going to discuss underlying dynamics in a simple, like this kind of dynamic, this outbreak. So the first thing is exponential growth. So exponential growth is natural if you think about one infectious person infects more than one. Let's say for 2 people, then the number of infected people becomes 2, and then again 4, and then an 8. So you have an exponential growth. But the number of cases doesn't grow indefinitely. It's bounded by the number of infected people, number of susceptible people. So if you look at this figure, you start from 1, becomes 2, and 4, and becomes 8. So you see an exponential growth. That's what happens in the beginning of the outbreak. But once it reaches, as the disease spread, there will be a situation point where you are infected more already infectious people, already infected or recovered people, so that you generate not many secondary transmissions. So there's a point where one infectious person generates less than 1. Then if you think about it, just like in exponential growth, you see an exponential decay. So the result will be epidemic ends, even susceptible people are still exist. So again, if you look at this diagram, this caricature, you see that starting with the 8 people, and then you infect less than 1, in this case 0.5. So you become 4 people in the next step, and 2 and 1, the epidemic ends while you are still susceptible. So this is the basic underlying mechanism, what's going on in an outbreak. So in the beginning, you have infect more than 1, you have an exponential growth, but then susceptible people becomes fewer and fewer, and infectious people less than 1 at one point, and the epidemic ends. Okay, so once you understand the basic dynamics, now we are ready to tackle some of the basic important concepts in infectious disease epidemiology. One of them is basic reproduction number. So it's defined by average number of 1 case, and you can sense that there is a threshold. It has to generate at least 1 to generate any sort of epidemic outbreak. So it's below 1. If you look at red line, you don't see an outbreak. So it's over 1, you see an outbreak, but higher or not will generate more rapid growth and higher case. basic reproduction number is important because it actually provides further information, such as, we're not going to describe details of this one, but it also provides final susceptible proportion or final epidemic size. It also provides vaccination, minimum vaccination coverage, assuming perfect vaccine. So if you look at this equation, if you say you have R equal to 3, then you have protect at least 2 to generate minimum at least 2 to protect an outbreak, so that the actual secondary transmission is not exceed 1. So one other important concept is herd immunity. It's an indirect protection from when it happens in a large percentage population has become immune to an infection. So if you look at this figure, you have 3 different colors: blue, red. Red becomes who are infected, so the infection spreads in a population in an unmitigated setting, and you infect just a few people. Then the infection spreads, but people who are vaccinated are protected. But that's it. You don't protect others. But if you protect sufficient number of people, then actually you can protect like these people who are not vaccinated but still protected, because this person's neighbors are protected. It's contagious disease, so if your neighbors are protected, and you're protected as well. So the idea, one thing you can learn from this one is we don't have to vaccinate all entire population to prevent an outbreak. So now we move on to actual models. So one of the most basic models is SIR. You divide the population into 3 different classes: SIR, susceptible, infectious, and removed. And the model describes how the number in each class changes over time. And you have defined two flow or rates. You have mu-SI and mu-IR. Mu-SI, the transition rate from S to I, is also called force of infection, or mu-IR is also the rate of recovery. In the next slide, you can say that it captures the characteristic growth and decay of the number of infectious individuals. One of the most common implementation framework is differential equations. We're not going to describe details in this equation, but let's look at the susceptible number of infected people, since that we have been looking at so far. So it's number of infected people influenced by the current number of infected people. That can, if you understand the rate of change in differential equation, they can induce a exponential growth, but then it can here the fraction of susceptibles to always less than 1, so that it can limit the growth of infectious people. So there are 2 parameters in this equation that are unknown. It's beta, is often called transmission rate, and it can be divided into 2 components: number of contacts per unit time, and transmission probability between S and I, susceptible and infected, per contact. So mu-SI, force of infection, can be defined this way. And also there's another parameter is gamma, that's rate of recovery, or one of gamma is the duration of infectiousness. So if you assign some number in beta gamma and solve these equations, you see, if you look at red one, if you characteristic equation and I, you have a rapid growth and then decay, while susceptible people are being still around. So this is a simple SIL model. But as we mentioned in the beginning, the simple SIL model doesn't provide sufficient answers to complex real-world questions. So we can extend the model to include more realistic details. One aspect is demographics. So the way we extend the model is to add arrows or add additional boxes, meaning additional compartments. So in this case, we have a flow in of susceptibles. It can be migration or birth. Let's say this is a birth. You have a birth and death process, so demographics. So the equation can be implemented. You can look at the equation later. One big difference between closed compartment model from the previous slide is that you have now an endemic situation where infectious people are around all the time. You're not going to die out. So in a closed population, it dies out, but in an open population, since you are a constant source of susceptible infectious people around all the time. Another aspect is age-specific contact. So this figure shows the data during COVID-19 period. So this one, if you look at the diagonal line, has the highest intensity in the diagonal line, meaning the same age group mix with the same age group population, right? If you look at the 10 to 14, they mix with relatively stronger with other 10 to 14, you call them 10 to 14, and compared to other, let's say this one is 0 to 4, 5 to 9-year-olds, right? So you have this is characteristic pattern common across all nations, right? You have a mixing between 0 to 4 with 24 to 40. There can be parents and child mixing. So this is a real phenomenon, and it happens in COVID-19. The intensity becomes reduced because of social distancing or lockdown, but then pattern remains. So we can include those. Again, the idea is adding additional compartments and adding additional arrows, flows. So then let's suppose the S1 being susceptible in kids population, it can be influenced by infectious people in the kids, but also infectious people in the adults population. So you can look at the equation later. Another aspect in COVID-19 is pre-symptomatic transmission. This data shows that, as I discussed in the beginning earlier, that negative serial interval meaning can happen only when pre-symptomatic transmission happens. So infectiousness profile happens before symptom onset. You have so this can be implemented. Another set of data show that if you look at out of 40 transmission pairs, quite a substantial proportion indicates that transmission happens before symptom onset. So that our understanding of the transmission reproduction number comes from about 0.9 out of 2.0 is a pre-symptomatic transmission. So this one can be included in the same fashion. We saw that this figure earlier. So you add additional column like you have additional boxes, pre-symptomatic, asymptomatic transmission. Then you assign transmission pathway from A to S and also I to S. So in an equation, if you look at that later, but just briefly, the transmission happens S becomes I by I or P and or A. So the same strategy will apply, so you add more boxes and more arrows. Another thing we can do, another aspect of the real phenomena is people's movement. So this is one of the studies of COVID-19 again. So this study captured the people's movement by, assigning the SEIL model at different census block groups. It can be residential areas or certain areas. You have a different transmission model, but then these models are linked together by a points of interest. It can be some areas where people mix, such as mall or a park or a stadium somewhere. So I'm not going to go into details of the equation, but then same, you add more compartments and more arrows, which can later be implemented in equations. Another aspect of vaccination, it's the same strategy applied. You add more states or class or compartments, and then you add arrows, which can later be implemented in equations. Then you write down equations, just like we did that before. Okay, finally, there are other aspects of ensemble models. This is important to understand because it shows that what models are like. Models are like a somewhat limited, it can only capture limited aspects of the reality. So model-based projections often disagree. That's perfectly normal, as I mentioned, because models are limited aspects only captures the limited aspects of the reality. So then our strategy will be to make best of the diverse projections of multiple models. In a previous setting, flu epidemics, it has been shown that more better predictions than a single model. So it's actually being used in the current setting, the CDC, to make a projection on natural weekly death. In different colors represent different modeling groups or different models. So then they have all different projections, and you combine these to make a projection. It can be a new weekly death or total death. In summary, so we have so far seen the mathematical model is a mathematical representation of mechanisms how infectious disease spreads. Differential equations are one of the simplest format you can implement to model. Models are useful. They show a likely outcome of epidemic and also can show the potential impact of intervention strategies. Simple as models may not provide sufficient answer to real questions, but then they can be extended to include real world aspects such as demographics, natural history of infections, age-specific contact, spatial movement of vaccination, movement of people and vaccination, and more. And finally, model captures a limited aspect of reality, and model-based projections therefore may disagree. That's perfectly normal. Ensemble projections may make better predictions by combining projections from different models. It can be provide better predictions than predictions based on a single model. Thank you.
Thank you, Dr. Kim, for a very informative presentation. Dr. Kim joins me here for a Q&A session. We have a couple of questions from the audience. We'll start with the first one. So the first question is, in Africa, modeling has had projected the likelihood of high COVID-19 caseloads. How do you explain the sharp contrast of actual numbers to what has been projected or modeled?
Okay, the first question, I think it's a very important question. Probably I'm not be able to provide the full answers. There are many dimensions, I think. I believe, first of all, I'm not sure which model, but most models actually predicted high caseloads, but models and realities always disagree. In this case, there are, I didn't look at the specific model, but there must be some assumptions about when the case introduced and how people mix each other. And also, there are, aspects of a model can be wrong. But then there are aspects that data can be wrong too. Mm-hmm. So especially COVID-19, we know that actual reported cases is just a tiny fraction of the actual potential case numbers because of the symptomatic cases and because of the limitation of the surveillance system in the country. So there are at least a couple of dimensions that we can think about.
Thank you. And then the next question is, how do you think modeling would have a place in determining COVID-19 vaccine allocation, and what would that be?
Let's see. Meaning that, so is it asking whether it's modeling can be useful in terms of, I would interpret that as modeling can be useful in terms of providing, designing vaccination strategies. So yes, I believe so, and that's what these days most scientists or policymakers think about, and WHO itself actually organizing various modeling groups to think about vaccination strategies, to those versus those interval. There are many questions are being answered, and many countries, in Korea too. So I think yes.
Thank you. The other question we have is, and relating to infections which might have different origins, for example, in terms of incorporating environmental and animal infection, given zoonotic origin and environmental spread might have different roles, and how would that be considered in modeling?
Okay, that's a good question too. Okay, environmental modeling. So as I mentioned in the presentation, one way to add more reality is simply adding arrow and boxes. So the environmental transmission has been already included in many modeling efforts that I know. So just adding environmental reservoir, whether virus can sustain, or it could actually more common in bacterial transmission, something like cholera or typhoid fever. They have explicitly included water reservoir in the compartment, and you assume that how many bacteria, do you say titer or no? Maybe colony forming CFU or something. So how many bacteria needed to make an infection? So you make a series of assumptions how people touch or contact with a reservoir with the bacteria or virus. So animal infection probably be the same, but it's also depends on the modeler's imagination. But if you can simply extend the one that we have introduced today, it's just simply adding more compartment for animal. So we have a separate compartment for animal, but then you can assume some sort of contact between human population and animal. So you can extend, smoothly as we add more age groups or as we did before.
Thank you so much. Those are the questions we had. We have flagged from what the audience asked, but Feel free to go to provide responses within the Zoom Q&A platform if there are any unanswered questions. And thank you so much for joining us for this live Q&A session.
Okay. Thank you.
Next, we will have a closing remark by our Director General, Dr. Jerome Kim, Director General of the International Vaccine Institute.
Good day, and thank you to everyone for attending the 20th Annual Vaccinology course. I hope that you've learned something during this course about the complexities of vaccine development for global health and the reasons that we do it, how we do it, and why we do it. Again, I'd like to thank our sponsors, GSK, Janssen, the National Vaccine Institute of Thailand, RAFIS, and SK Bio for their support of the course. This has really been a remarkable event. It's the first time the course has ever been offered virtually. We had 6,964 people sign up for the course on the first day. 3,634 people actually attended the course on that first day. And again, by comparison, in the entire history of the course, we've had about 1,500 participants. So this has been a record year in many ways, and I hope that it was valuable and productive for everyone. And we are very eager to hear your comments on the format, the presentations, and how we could potentially improve the course in future versions. I think one of the other remarkable things was that we had over 400 questions on some days, and that's really incredible. Having sat through vaccinology courses in the past, we never have the ability to get to all the questions. But I think on the virtual format, we were able to do that, and I hope all of your questions were answered. Feel free to send additional ones. Remember that the talks are available on YouTube starting at 3 p.m. Eastern Daylight Time based on the United States. And also that slides will be made available if the presenters accept this option after the course is over. Once again, I'd just like to recognize the people at IVI who were responsible for organizing this year's course. Dr. An Wortel, the course director, Miwa Ka, Satyajit Sarkar, Jaesung Yang, Ondari Mogeni, Berkne Tedessa, Zenaida Mohares, Gyong Pang, Youngmi Cho, Taekyung Byun, Ari M., and Cindy Park. We really want to thank you and congratulate you for executing this virtual course for the first time extremely well. I hope that this course has given everyone a passion for what we do. All the people at IVI feel very dedicated to our mission, which is to discover, develop, and deliver safe, effective, and affordable vaccines for global health. We really feel committed, and we hope that during the course of the presentations, some of that excitement was related to you. And again, if you have additional questions or if you want to come and visit IVI or if you'd like to come and work here, please let us know. Thank you.
Okay, thank you so much all for joining this vaccinology course, and we hope to see you in person next year. And thank you. We close the session here.